hello to everyone. Here I'm very happy to open this hybrid event, Biotech Panel of the Smart Conference. During this day, a lot of our partners and collaborators, friends and colleagues will come virtually or in person in order to see us and to work with us. So now, in order to start this as official as possible, I want to give the floor to our Vice Rector, Professor Khaled, to say some comments.
Can we use Bulgarian?
No.
No, English. So, dear colleagues, very good afternoon. I would like to thank you for joining us in this conference. Varna Free University is very proud to be able to host this conference today at this wonderful place. Unfortunately, during times of COVID-19, we cannot see the whole opportunity, to work in our university. And also we encourage all conference members to work in safe environment, and because of this we have this conference in this dual forum in this room and also in Google Meet. The topic is very important, the topic is very interesting of the conference. And also we have very interesting participants. I'm sure that we can learn a lot from each of you, and also that in your cooperation you will end up with good ideas and good solutions. So I would like to welcome you to this conference and to wish you a very fruitful conference, a very fruitful work. Thank you very much.
Okay, so first of all, I want to present all the people who are here and all the people who are online, and when some new person is coming online or in person, I will present him also. So here I will take this floor in order to think a lot. So this is Professor Stoyan Pavlov from Medical University in Varna, Professor Teodora Bakarjeva from our department, our Dean, Professor Tankova, Dr. Kordon from Kordon Consulting, Todor Dobrev, Head of Campus Company, Professor Tonchev from Medical University in Varna, and Vladimir Vatev, one of our alumni students who is also entrepreneur and is the manager of the Biobuda company, and our colleague Mr. Neil from our department. So these are up to now the people in our room, and from our people online, we have here the head of Biocluster in Bulgaria. We have Christina, so I will be very glad if you say some words later on. So here is also Professor Semyonkin from Sibir University, our new partner. Here are a lot of students who are new and old ones participating in our research groups, like for example Todor and Mark, and new people like Alexander, Boris, Diane. And here is also Petra from high school students, Katerina from Planteral company. Here is just coming Emil Tsankov, the new head of ICT cluster in Varna. And also here is Mark from USA. Thank you very much, coming from Columbia University. And Radko Mishev. So these are the people who are now at this moment in the meeting. Thank you for coming, and I want to tell you that I'm going to ask you some questions, and I'm going to try all my best in order to do the collaboration between and communication between all the people who are here in the room and also people who are online. So our event will be provided on the program that we gave before, and we have a lot of presenters. They will be mixed here in person and online, and a lot of people will come on exact times. That's why it will be very dynamic. And according to the topics and our mood, I will give some breaks in some periods of time according to the circumstances and our emotions and so on. So here the first thing that I want to mention here is that this year actually this panel, this conference is gathering topics that we've been discussed in the last conferences, but here they are some kind of summarized in a different way under the umbrella of the biotech industry and practices. So we want to show that a lot of our research could be used and could be a good and the benefit of this type of industry that is extremely perspective, because it concerns our health and good and healthy lifestyle. So this is the conference website, and also all of these presentings and recordings will be shared with a lot of people later on and in the Facebook event. So I'm going to the drafting a little bit what happens with the program now. So I'm going to say some words about ecosystems and their orchestrations. Then we invited Dr. Mario Guerra, who is chair in Transstem project in Medical University in Varna. And after that, we're going to say some words about practices in computational life sciences. Then it is a session about biomedical image analysis, and here again we're going to share the experience of our ecosystem, but also we invited our prospective partners from Germany in order to share their experience. And they will be shared two types of experience, and one of them will be from one startup company. Then traveling to Varna at this moment is Jordi, who will represent his experience in his startup about image analysis. So here we did our best in order to combine research practices, educational practices, and also business practices. And as for the neuroscience session, the last one, it will be sharing some neuroscience interdisciplinary ideas, research and practices. And Dr. Cordon will share here his thoughts about and knowledge about genetic programming. In this panel, we decided to put some important projects that are developed under they are actually active projects in all of our partners, and these projects will be in 3 areas that are in connection with this biotech panel conference. And at the end, it will be a very pleasant matching session with some discussions between the researchers here in the room and young researchers outside, and also later on huge just party with a lot of our business partners, our colleagues from universities, and our young researchers with Ulina Chavda and other people you will see there. So this is the program in brief. Now I want to share some thoughts, just some thoughts about research and innovation and entrepreneurship ecosystem. Because we claim that actually we want to extend and enhance the existing ecosystems in research and in entrepreneurship, and somehow to combine them, and not to cover and to copy existing one systems on regional level, because we know that everywhere in the world it's different. So we are taking under consideration the requirements and our style around and everything with our partnership, and that's why we did some some interesting things here. So these are some focus areas of special interest groups in which we are combined the people here somehow, and a lot of us are belonging actually to different ones. So we have people who are students from medical university who are going to do some different segmentation and labeling, preparing images for neural network analysis. We have people who are ready to do different type of research in the area of neuroscience. We're going to discuss this later. We have a special interest group here in the university that is about developing new neural network designs, and a very good perspective and very strong special interest group in textual analysis from different people, from different institutions, from our both universities and other researchers. And the last one is a special group about bioinformatics. So up to now, we are taking this as examples because it's very important that our strong community here and ecosystem is coming from the open practices between joint efforts between experts in different universities, interdisciplinary research. And these are actually the focus areas of our research and innovation tracks that we're going to do this year. And it's more convenient for us to put this on the academic year, you know why. So the first one is biomedical image analysis, the second one is computational life sciences, and the third one is neuroscience. And on this way, actually, we put an organized conference on the content level. So why this is important? Because this is a very interesting approach that we know that it is innovative because it's not happening everywhere in the world. And this gives us the freedom to do open practices and to establish new things very fast. Because we know that sometimes it's difficult to do something similar in university. But it's very important actually that we are helping to our research communities in the universities to go faster and to scale up faster in new directions and to do some kind of very dynamic data speed. So these are people and actually profiles of people who are participating in all of this. These are not only researchers from research labs, research institutions and academics, but also students from universities, high schools, PhD students, and some people from companies. So we want to extend all of this in new directions and new dimensions. A very interesting thing is that some of us who are here in this room now, we're working on the evaluation and self-evaluation of the ecosystem itself. That means that we want to prove that this system is sustainable because our senses and we think that it is sustainable, but actually it has to be proven that this is on this way. So that's why we're evaluating our ecosystem on 3 different levels. The first one is about research and articles. It's very clear artifacts about this. The second one is about educational activities. That means that somebody is teaching somebody else, and it's not necessary to be a lecture course in the university. It could be something additional as educational practice. And also the last one is entrepreneurial practices. That means that somebody is paying for somebody else in order to do this, or exchange some goods and activities in order to help each other. So at these 3 levels, actually, graphs of relationships we're evaluating now in different ways. And the first article we've already prepared about using social network analysis methods. But now it's coming to predict how actually this ecosystem will behave after involving and attracting new people to come. And also especially after the connection between different ecosystems in other regions. And the second approach that we've chosen, this is a multi-layer graph methods, because we find that it's very suitable for this. So these are the things that we're doing now. And here is some kind of movements of artifacts that proves all of this. So the first one are very important new educational practices that we are doing, and this beautiful project will be represented later from Professor Tonja. And this is extremely important because we are trying all of this as informal communication work and perspective work that we see in the future to formalize somehow. So this project actually is one that we applied under Erasmus Mundus design measures, in which actually we want to connect the three different ecosystems. This in the region of Varna and in Bulgaria, this is in the region of Romania and Cluj-Napoca, and this in the region of Hungary. So all of these actually there are some kind of special benefits in all of these ecosystems, but combining all of this and developing and designing a new program, a master degree program about computational life science will be of huge importance for all of us. And it's not because only the result will be the next master degree program. The result actually is that we're going to do a community that is more composed and work together better, with better team, that is international team. And it's not only international, but actually this team involves different players in all of this game, because here are the IT business, the university with IT specialties and medical universities, and also municipalities and so on. Because all of this as research is very important to be connected with the government policies. And because we really believe that all of this that we're doing is somehow helpful for our own health, not only for our own research. So this is in brief, and now I want to go in the program in the next presenter. And in order to do this, because it is a little bit earlier as usual, I want to give the floor to some of the people who are here to say some words. And let me choose some people here. So, Christina, can you hear me?
Hi. You meant me, right?
Yes.
Hi. Pleasure to be here. Thank you for the invitation. I'm in charge of the life sciences area, and also co-founder of the Cluster of Artificial Intelligence area. So I'm sure that today with the colleagues we will find a lot of way how to cooperate with the challenges and opportunities around the world. So I'm here to answer all of your questions and of course, discuss how we can cooperate in the future.
Okay, may I ask you to say some words about the profile of the members of the biocluster in Bulgaria, and what do you think about biotech industry in Bulgaria and biotech startups?
Sure. Okay, so Health Life Science Cluster Bulgaria was founded 4 years ago by the companies and organizations which are filling up all the fields of the life sciences. Started with the companies which are dealing with the 3D discovering of new small molecules, novel molecules. We have companies dealing with the human print 3D tissues, and of course we have companies dealing with the genetic examination. We have patient organization, institutes and university to support this matter, but also we providing and having in our cluster ecosystem companies which are covering the fields of the food tech and agri-tech, because we believe that they are also a very interesting innovation that can be brought on the market. As a cluster, we are part of the Eurobio. Actually, from the region, we are the only one presenter, so we can support, of course, the connections and any kind of activities that Eurobio can support. Also, we are part of the Deal Region Association, CRB, also a huge organization which having in their focus health tech, medtech, digital health. And as a representative of the AI Cluster Bulgaria, we are part of the European AI Forum together with the strongest and biggest association in the field of artificial intelligence from France, Austria, Germany, Poland, Hrbatiya, Slovenia, and here that our Bulgarian ecosystem actually bring on this Whistler ecosystem is to find a way how to combine biotech, health, life sciences solutions with artificial intelligence as a fundament of
Okay, so I'm asking about biotech industry in Bulgaria and startups, because I know from you that a lot of companies that are participating in BioCluster, actually they are not companies from Bulgaria.
Actually, all of the companies and organizations that are participating in our cluster, they are Bulgarian ones. Of course, some of them, they having, of course, markets abroad. And this is quite normal because unfortunately in Bulgaria we're suffering from the lack of capitals and venture investment when it comes to deep tech. And as you probably know, because I see a lot of scientists here among us, it is quite hard, not only in Bulgaria but all the region of Eastern Europe, to find investors who actually understand and can evaluate and validate deep tech innovation. So very often, unfortunately, a lot of the startups supposed to discover and even transfer their IPs into other countries in order to attract such an investment. Actually, right now, the cluster is working proactively how to fill this gap. We start working together with Oxentia, which is the innovation company of Oxford, to support us in order to build venture build studios as a model that can provide and fill this gap. When it comes to the sector, unfortunately, we don't have exact data because we started already a mapping of the ecosystem, but still it will take a little bit time. There is a huge amount of companies which are claiming they dealing with biotech, with artificial intelligence, but it's not correct. So we need time to, of course, to evaluate all of them in order to support them to get stronger numbers.
Okay, and I have one final question from my side. So this is the following. So you mentioned a lot of companies with common interests like ours. So when actually are you going to meet us with them?
Again, I'm sorry, but I can't hear very well the question. Can you make it louder?
I'm asking the following. You mentioned a lot of companies with common interest with our ecosystem. So I'm asking you and provoking you, when are you going to meet us with them?
If I understood correct, we're talking about again for the profile of the companies of the cluster or the ecosystem, international one that we can contribute by introduce to Bulgarian ecosystem, but of course with the biotech organization, right?
Okay. Okay, thank you.
Okay, thank you. Sorry. Thank you.
Okay, so now I'm just going to check the people who are here, and so I'm using several more minutes before the next speaker to come here in the meeting, and I want to ask Professor Semyonkin, are there something interesting in your ecosystem, in your community, concerning biotech industry and connection with research that you want to share with us?
Sure. I can share a little bit more about the possibilities of cooperation between the Bulgarian ecosystem and European one. As I mentioned, we are part of the Europe Bio right now. There is a very interesting projects and how to say consortium that are established right now. Some of them are connected with the digital innovation hubs in Europe. As a cluster, we are part of the DIFNet Europe that provides us direct access to 35 medical digital innovation hubs across Europe. The projects that right now are most wanted, if I can say so, are in the field, of course, of the drug discovery when it comes to orphan disease and uncurable one. For example, one of the projects that right now some of our members and Western itself is working is it's called the genome, which is 110 associated gene mutation, and found the 110 small molecules to cure all these gene mutations associated with the breast cancer. So there is a colleagues which are interested of this topic. Of course, we can support this. Of course, probably you know Bulgaria is part of the 1 million genome program. So there is a very interesting project that all of you can participate. And yeah. Biotech is ruling right now as a project, as inspiration, as networking in Europe. So, yeah.
Okay. Thank you, Christina, very much. Thank you.
Thank you.
So I wanted to ask some other person from another point in the world to share some information about possible collaboration about biotech industries in the area of research and entrepreneurship. So if you are ready to share something, please raise your hands or write in the chat, and I will see. And my colleagues prompt me to give you the floor. So this is some kind of a short and brief information about the second topic, in which our ecosystem has some kind of experience and some efforts. And this is the topic that we put this general title in Silicon Life Sciences, and it is actually connected with the topic about computational life sciences. So here in this area, we tried a lot of things to do, and the first one was one course last summer with lectures that you see here, Professor Черноризец and Professor Pavlov leading this course about DNA, RNA, and what happens there in bioinformatics, and basic terms and concepts in one of the software companies in the city of Vantarau, because they wanted to teach somehow and to give some knowledge to their staff in this area in order to go in this area of preparing the software for biotech industry. So here also there are a lot of interesting things about courses that we did last year every Sunday with Professor Черноризец, Professor Pavlov and me about Silicon Life Sciences and a lot of topics. And the very good result in all of this is that we have recorded videos in English of everything that we think is important in this area nowadays, and it deserves to be observed, researched, and studied for business purposes. And after this course last year, we actually decided to do something different and to organize such kind of Sunday sessions, educational sessions in different way. And this different way will be in such a way that we're going to put one research-focused problem that is a real problem, and we want to go and to search for a solution in this problem. And during searching for a solution and doing this and designing this research, we're going to teach people who are going to together with us and to come with us in order to do this and to give some knowledge in this area. So this will be some kind of different approach for organizing the course that we want to experiment. And after that, Professor Черноризец will share an idea of all of this. So also there are very prospective partners of our community and our colleagues from Medical University, and we are gathering everything that is possible and that is helpful in order to not only to receive, but also to organize the most important knowledge, especially for people in interdisciplinary areas, and for such a community in this bioinformatics unit in this Italian university. And also here we have this privilege to represent our work with the support of Christina Eskenazi in one special event. And it's very good, and I want to thank her very much that she gives this opportunity to all of us to share our experience in order to popularize what we are doing actually. This year also, for institutions, the Varna University, Medical University, High School of Mathematics and Quantaro Company are officially organizers of the first DNA Day in Bulgaria. And we know very well that we're going to do this next year too in April. And it's very important because we put all of our expertise in all of this day in order not to repeat everything else that people around the world are doing, but to do something different, to combine all of this from biological, computer science and art together with music and art and fine arts and everything that was very, very good and interesting and very, very helpful for all of us. And it is very important because it engages people to pay attention in all of this and to do more science and to love science very much. So this is the Facebook event, and you can see the videos there for a lot of presentations. So I will finalize all of this that it's a new trend in Bulgaria to have a national website of open science, but actually open science is not new for us because we have our open practices for doing science in different way, and we think that it will help us to scale up in different directions and with different speeds. Okay, so now what about our next presenters? Are they here? Okay, now I see here that Dr. Mario is here. So before giving him the floor, I want to give the floor to Professor Tončev to represent him. Please.
Hi, Mario.
Hello, I'm here. Hello.
Hi. So let me say a few words about Dr. Chiguera. He's of Italian origin. He has spent many years of his study after Italy, after graduating in Italy, in Switzerland, in the United States. And before coming to Varna last year, he was for about 5 years a group leader at the International Clinical Center of the Maverick University in Brno in the Czech Republic. So how did we come to Varna? That was and it still is, a European project which is called ERA Chair. The European Commission has introduced some 5-6 years ago a type of projects that aim at attracting a leading international scientist in a given field, any field of science, to the countries which are at and below the average of the EU. So this is a kind of call where countries like Bulgaria, Czech Republic, Slovakia compete between them and not with Germany, France, or England, or Spain, or Switzerland. So in this call, one has to promote the creation of a whole team in a given field of science, and this is stem cell biology in our case, which is led by a leading international scientist, and this is Dr. Vinci Guerra. He was elected with a international job call announcement. Around 20 applicants have applied from all parts of the world. And then the next step is the creation of a team around the ERA Chair and a number of activities which are both educational and of course scientific in this field of science, which is the project all about. So now we are at the stage of compiling the team. The leading postdocs of the team are now in Varna. And so we are at the stage of also bringing some PhD students and part-time postdocs, and by the end of this year I hope the team will be compiled. Just to give an impression of the high standard of science Dr. Guerra has had and is having in his career, is to mention that he was elected first in with a number of candidates. About 20 candidates applied, as I mentioned,
and for example, his age index is around 45. So we are very pleased to have Mario here. For less than a year since his arrival, we've had a number of very nice collaborations initiated. Galina mentioned one of these with his colleagues in Italy at the Sofferenza Clinical Hospital, the Research Hospital, and the Mendel Genetics Institute in Rome. But there are a number of others which are going on, so we really hope that his stay in Varna as air chair will be very fruitful, not only for the local community at the university and at the Varna region, including the local biotech and the bioimage analysis group, but also even in a broader regional level, including adjacent countries at least, such as Romania, Greece, or Turkey. So thanks, Mario, for participating, and we are looking forward to your talk.
Hello, good afternoon, everybody. Do you see me? And do you hear me? I mean, do you see the presentation?
Yes. Yes, the presentation we see.
Okay, and you hear me as well?
Yeah.
Okay, great. So I'll start, and I'm going to thank Anto for the kind presentation and also the organizers of this event. So Anton introduced me. So what I would like to do today is to present you the structure of this virtual department that we created with the support of the STEM project. So the department is called the Stem Cell Biology Department. So this was a slide about myself. Anton already said a lot. So basically I am Sicilian, from Sicily, south of Italy, and I spent the last 20 years in several European universities. So at the Milan in Geneva, at the EMPN in Germany, and the UCL in the UK. Then I was 5 years in the Czech Republic, and since less than a year, I joined the University of Varna. So the goal of this newly created department, supported by STEM grant, is a 4-year project, and it has several goals. The first one is to strengthen the scientific excellence in the Medical University of Varna in the field of national stem cell biology. Also to transfer this offer of innovation potential of Medical University of Varna. There are also other goals such as to develop the regional policy about the stem cell biology, which is not developed, to extend collaborations to national and international partners in order to raise more money, especially the European Union funded goals, and to liaise, so to create strict cooperation with the clinical units that are present at the Medical University of Varna, especially in the hospital, and finally also to disseminate the results of this project at the regional, national, and also international level. So we have one motto, one phrase, that is our symbol. So we want to realize full educational and participatory potential of stem cell research in this area of Europe, sea coast of Bulgaria, to drive a new era of biomedical development, growth and productivity. So this SCBD, as we call it, the Stem Cell Biology Department, officially is one of the 6 departments of the Research Institute of the Medical University of Varna, and this fits very well with the scientific priorities of the university in terms of regenerative medicine. So the department is spread or scattered through 3 different locations in Varna. One is the main building of the university, where the rectorate is in the center of Varna. Then there is the Faculty of Dental Medicine and the University Hospital Santa Marina. And as I speak, we are organizing the establishment of a new central laboratory in the main university building in order to have all the equipment and all the facilities as a main European project, we might be familiar with it. These are organized in work packages. So there are 9 work packages. Here you see in green the one that we already realized, we already achieved. Basically, is the fact that I was brought here, and also the recruitment of the team, and the establishment of a strategy for the realization of the project. While others from work package 4 to work package 8 are ongoing, and it will be designed. The results will be realized and are already realized in the next 3 years. So the principal investigators of this project, there are 5. One is myself, another is Professor Tomko, Professor Kristina Grubcheva, Professor Resnik, and Dr. Chervenko. And our interest or expertise are really crossing to each other, because I'm specialized in cancer stem cells and also induced pluripotent stem cells. Dr. Resnik and also Dr. Chervenkov are specialized in hematopoietic stem cells. Dr. Tomchev team works on neural stem cells, and Professor Grubcheva developed an interest in limbal stem cells, that are the stem cells that are located in the eye, in the corner. Also, we take advantage, so the project supports also administrative personnel inside the University of Varna. So we have Mr. Nicola, Mr. Donchev, Mr. Ivanov, and Mr. Markovska that help out a lot with the organization of administrative matters, also IT matters, and Mr. Markovska help us with the dissemination via television. So the department of communication is also very, very important. So this department is actually really part of the university. It's not like detached from university. In fact, the PI have important roles within the university. So Professor Gupta is the vice director of the university, the Faculty of Medicine. Professor Toncic is the director of the RIMO. And Dr. Resnik is the deputy director of RIMO. Also, Tomchev, Grubcheva, and Resnik is also teach at the university, so there is a direct contact with students who might be interested in working in the lab. And Resnik and Dr. Chervenkov, they also work in Santa Marina Hospital, and Professor Grubcheva is also director of the AL Hospital. So this project will last 4 years. So I'm here for 4 years, and we have a management board that meets regularly. We have a college of group leaders, so all the colleagues that I mentioned so far, and there are also, as I said, administrative and technical department. And we all meet regularly to implement the project. The project is also supervised by an advisory board. It's an international board that check that everything goes according to the plan, and everything also is developed regularly. This advisory board is composed by big professors, established professors in Israel, in the USA, in Germany, and in Greece. Also, what I would like to tell you is that there was the need of a project of stem cells in this part of Europe. In fact, in 2017, with the arrival of Professor Resnik from Israel, for the first time it was done a pediatric stem cell transplantation for hematological malignancies at the University of Santa Marina. And the Professor Resnik is the coordinator of the program. And also, if you look around, so what people are doing around Varna in Bulgaria or in close Romania, there are only few institutes that work on stem cells. I list them here. So there is a Profit University, there is an Institute of Biology and Immunology of Reproduction in Sofia. There is a society called the Bulgarian Society for Regenerative Medicine, but this is only a consulting body. And there's also an institute at the Nicolae Simonescu in Bucharest. So, but this institute, they work with very specialized or sectorial aspect of stem cell biology, which concern, for instance, reproductive biology or bone biology, but they don't have a comprehensive approach towards stem cells research. In fact, I think that what we create in Barna is the first institute that try to address several aspects of stem cell biology at the same time. So, hematopoiesis, genesis, corneal cells, and also IPS. And we have actually a portfolio of experimental models and techniques that place us in a position to create a pandemic center of excellence in stem cells in the country. So within this macro area, so the macro area I define hematopoietic stem cells, neural stem cells, solid cancer stem cells. We plan to use preferentially innovative approaches. Innovative approaches you find on the bottom of the slide here, which are epigenetics, distal biology, 3D bioprinting, and artificial intelligence. So epigenetics is my field of expertise. So you know that in the nuclei of cells, gene expression is regulated by the compaction of the chromatin, because the DNA wraps around octamers of histones that are basophilic proteins, and if the chromatin is more or less compacted, this will basically regulate if some genes are switched on or switched off. So this is of fundamental importance. I will tell you a few words next. System biology is also a key area, because nowadays every laboratory, and also in Barna, and I think in other laboratories in the country as well, researchers generate high throughput data. So they generate a larger quantity of transcriptomic data, genomic data, proteomic data, lipidomic data, and these data are harder to analyze with the conventional techniques. So, system biology approaches, which are basically softwares that are dedicated to the analysis of, simultaneous analysis of multiple omics, it's possible to identify new targets and new data that can be verified and analyzed also in vivo setting. And another area is bioprinting. So, you know, bioprinting is becoming absolutely important for the generation of prosthesis and also model in vivo environment. And in Barna there is a very good and expensive 3D bioprinter, and it's possible to use machines generating scaffold in which we can create stem cell environment and study their biology. So this is also a strength of the medical university. Artificial intelligence is very fashionable, everybody talks about that. In the field of stem cell biology, it's possible using pictures of the cells to run the softwares for informatic approaches to basically describe the phenotype, so to describe the differentiation of those cells. In fact, it was reported recently that the machine learning, which is a particular type of artificial intelligence, can predict the Leuker stem cells, because this software can analyze thousands of 3D cell images and can actually tell which of the cells are differentiating into the cells or the stay under embryonic state and so on. So this is something also that we are looking into. Why is this important? This is important because stem cells is a very controversial field. Some approaches failed when trying to translate them to the clinic, but some of them succeed. So this was a very recent paper last year showing that actually by reprogramming the cells in the eye of a blind mouse using epigenetic approaches, apparently it was possible to restore the vision of the mice. So the literature is full of exciting examples of basic research that, if translated to the clinic, can really make an impact. So I have described, let's say, our intents, what we want to do, but we're already active, and we publish a few papers in a year or so. I said that my expertise is in genetics. In fact, I study an histone called microH2A. It's a big, large histone expressing all mammalian cells, and this histone regulates apparently neurogenesis. And we published this paper very recently. So the absence of this histone actually boosts neurogenesis and improves social behavior and memory. And there was also another work where we studied this histone in the differentiation of the somatic cells, so the cells of our body, not reproductive cells, into induced pluripotent stem cells, IPS. And by playing around with the expression of this histone called microH2A 1.1, we have shown that this is a paper which has been accepted for publication, that it's possible to modulate the DNA damage repair mechanism that occurs in the cells that are important for the differentiation of the cells, and we can actually improve the reprogramming efficiency of IPS, which is a very low efficiency phenomenon. So it's about 1%, 2%, and by playing with the expression of this histone, we can double this efficiency. So, and we're continuing in this line of research in Boston. So this was my last slide, and thank you very much.
Okay, thank you. Do you have some questions to Manuel? Okay. So, do you have a sense of humor, Manuel?
Sorry?
Do you have a sense of humor?
I do.
Okay, please start another presentation, Manuel Joe.
Okay, what should I do?
You have to watch only.
Yeah.
We saw this picture of you in the internet, and we want to show you what type of things you miss because you are going to have a party in the park. So this is the park where we are going to have a party. So we're waiting for you coming to Bulgaria to do gardening and do a film. Okay?
Okay, I will be there tomorrow morning.
Okay. Thank you very much for participating in our conference. Okay.
Thank you.
So next in our program is Professor Черноризец. Trifon, are you here? Okay. Well, Trifon is answering us. I wanted to ask Mario, what do you say about the usage of all of this research that you are doing? For what it will be helpful for every person, not only for medical and biological researcher.
So, thank you for the question. I think that there are two parts of this project, the basic part and the clinical part. So for the basic part, I'm a basic scientist, I'm not a doctor, we create information, we create a knowledge that maybe one day can be exploited for trials. But already what Professor Resnick brought to Varna is very, very practical, what Dr. Cherbenkov do. So they study how to improve autologous stem cell transplantation efficiency to try to save the lives of people that have immune system compromised or they have cancer as well. So the project is interesting because it contains both aspects, the basic research and the clinical research.
Okay, thank you very much.
Yeah.
Okay, so what about Trifon?
Sorry.
Okay.
Can you hear me now?
Okay.
Can you see the presentation screen now?
It's coming.
Okay. So, I'm sorry for the interruption, but for some reason I wasn't able to hear you and understand it, but I hope now everything is fine. So, here is a short presentation of initiative which came over in the last month, I would say. So, as you can see, title is an initiative for metagenomics analysis of urban wastewater. So here is a short outline of the presentation. So first I would say several words about metagenomics. Metagenomics, what is it, why it is used and how it is used. Some words, this follows the next short talk on the next of the human microbiome, why it is important. And actually metagenomics is used to study the human microbiome. And the final thing is about urban wastewaters. Actually, it is a hypothesis that they may serve as a proxy for urban microbiome. So, what is metagenome or metagenomics? The term was relatively recently created in 1998, so that's the beginning of the new century. So the term actually contains the prefix meta, which derives from Latin and would mean in this context on high level of abstraction, or it is collective genomes. So metagenome is a collective genome. So actually the term represents an approach where we may characterize microflora, as used in the term used in the original publication, or microbiome, for their properties, this is the original idea, or their composition, importantly, by means of genetic analysis. And the most important thing is without culture. Why? Because actually many microorganisms which are present in the soil, for instance, in the original application, are present in nature, and some present in our own microbiome, actually came with culture with our current knowledge and our current cultural system. So actually, in this way, by genetic analysis, we are able to circumvent this huge obstacle and gain information for the structure, the properties. So this slide is inserted from the original publication where the term metagenomics is coined, and actually the original idea of the authors was to use the metagenome to find new chemical compounds and enzymes. So without culturing. So in the original approach, they took the soil sample. After that, they isolated large DNA fragments, cloned them into the body, and then characterized the chemical synthesized by these transformed colonies and looking for enzymes. Actually, this was just an idea. They did not show implementation in their first in their article. And on the right side, again from the original publication, this is the some chemical compounds that were identified and possibly identified with this approach. So next. So how metagenomics is done? So cloning was the initial approach, but currently with the advent of next or new generation sequencing, the usual approach characterized the metagenome from environmental samples or the human microbiome or other microbiomes is by next generation sequencing, and the two usual applications which can be found as ready-to-use services where you could send a sample and receive a result is an original approach was to sequence the 16S, the large ribosomal subunit ribosomal RNA genes, which is a gene sequencing by short reads, and the the next logical approach and the current most good approach is to shorten sequencing, to use shorten sequencing, or to sequence via shortly complete genomes or complete, well, it's not correct to use this term, complete genomes, but DNA isolated from crude material samples without enriching for ribosomal DNA or some protein. So it's a representation, they are complete genomes, but it's not complete genome sequencing. On the right side, you could see the characteristics of example service, which you could find. There is no targeting, so it's whole genome sequencing by short noun analysis. So you could detect bacteria, here, fungi, protozoal viruses. The lowest detectable taxonomic levels in this approach is species. We can and you can take microbial functions here, like influence on new checkpoint inhibitor therapy and so on and so forth. So importantly, with 16S RNA gene sequencing, we could only profile microbial species, and actually we cannot go to individual species level. We cannot a certain function, so it's a major difference between the 16S gene sequencing and short-run sequencing. So it is better. However, a major drawback that transport technology, including for microgenomic analysis, is that it is short-lived. And short-lived introduces ambiguity in genome data, which sometimes cannot be resolved. It's a problem for genetic testing, but here in metagenomics, it is even higher or bigger problem. So actually, what is wanted, what we would want is to use long-lived technologies, where we could, as you will see on the next slide, less ambiguously or unambiguously identify or assemble de novo material genomes. And in this regard, a crutch, or I don't know how to say, a patch regarding technology is introduced by a firm called 10x Genomics, where we are DNA barcoding of large DNA molecules before fragmentation. And after that, they are sequenced via short-lived technologies, but with barcoding, long-range information is restored. After by special analysis techniques. So it's a short read, but it's long-read right, because we could restore some long-range information. It's the good about this technology that it's available now, and it is relatively cheap and gives us more information. Another technologies which are available now is long-read by Pacific Biosciences, but these are more costly, so it's still not very plausible to use routine analysis. And the future is via nanopore sequencing, which is through very long reads of individual genomes will be efficiently and hopefully cheaply assembled by this technology. It is still not widely used due to still a bit higher error rate compared to technologies, where they are constantly improving, so hopefully soon they will catch up. So in this slide, I have shown the difficulties and the advantages difficulties associated with short reads in one hand, and the advantages of long reads on the other hand. So you can see almost like, so the black line in the center is the full genome of the reference, or the assembled, the full genome, let's say, the full genome. On top are short reads, which are, as you should see, they are identical. And on the bottom part are long reads with different colors. So you should really see that the short reads, due to the fact that they are short, they may map to different regions of the reference genome. So this is actually ambiguity. So you cannot discern the correct position of the short reads on the original genome. On the other hand, the long reads overlap with each other. So on one hand, they are uniquely mapped to the reference genome. And actually, another good thing about long reads is that due to the overlap, you could actually assemble the novel genome. This means that we could build a reference genome for organism, or in our case, microorganisms, without having prior information. So this is called de novo assembly. Or the usual approach with short reads is to use reference assembly, so compare each of the short reads with the reference genome. For the human genome and for genetic analysis, this is present because the culprit of the human genome coded. However, a major limitation of genetic studies is that we have reference genomes for a very small fraction of the possible control, which are present in our samples of reference. So this is I would like to highlight that this is a major obstacle, a major problem actually associated with genetic studies. So a few words about nanopore sequencing. This is a technology which I am very fascinated by, and I hope that it will become part of our everyday life soon. So it's a very simple idea. You can see on the left side a nanopore, a very small pore, which is present in this case in a lipid layer. It is has very slow opening in the middle, where just a single base can fit. So denaturated single-stranded DNA is passed through the pore due to electromotive force. So each base obstructs the orifice of this nanopore, so it creates a character which blocks the electrical current while it's present in the orifice. So this generates a characteristic current which is unique for each individual base. So it has the potential to be very efficient. With this actually technology counter that is a long history. Actually, the change with this technology is something more than 2 million base pairs, which is huge compared to the 100 base pairs which are usually sequenced with the secondary. And on the other side, and on the other hand, you could see the sequencers are very portable, so they can be brought to field. With this technology, for instance, viruses are sequenced in several outbreaks at the field. So, several words about the human microbiome. So it's a very, I would say, modern or fashionable topic during the coming years. So here are some short list and some basic facts about the microbiome. So importantly, our human microbiome counts of something like 100 billion microorganisms, which actually outrages the number of cells in the human body, in our bodies. So it's something like 3 times more we have microorganism cells compared to our own cells. So another fact is that microbiome contains something like 200 more genes compared to our human genome. It was kind of strange to know that the gut microbiome contains actually violate 3 kilograms and the skin microbiome is something like 1.5 kilograms, which is kind of unexpected. And importantly, what we know in the index, which was part of studies of the human microbiome, is that actually we know now that it influences health and disease in a variety of ways. And the link with the metagenome studies is actually that with this technology we were for the first time able to comprehensively profile the human microbiome, so we could get information about health and disease. So importantly, there are some animal models where germ-free mice were created in the laboratory with the use of antibiotics. So what we know from this germ-free animal models is that the organism cannot function properly without microbiome. In the first place, it's the immune system, so it assumes, so its normal function assumes the presence of the microbiome, but also the brain activities somehow change. So importantly, microbiome composition and diversity is influenced by different disease states. First known for disease where it is known that the microbiome perturbations are associated with hemorrhagic colitis due to Clostridium difficile. Well, I won't go into that, although it's very important. So on this slide, in short, we can see actually that the gut microbiome can affect the function of logically and obviously the intestine, but also the liver, the kidneys, pancreas, brain. So the whole organism depends on the gut microbiome. And on the right side of this slide, you can see different factors which influence the composition and diversity of microbiome and may thus contribute to health and disease. So it's our host genetics, antibiotics, very importantly, gastric secretion, the diet is very important factor, and so on and so forth. So I'm coming to the final part. So the original idea is to study wastewater microbiome as a proxy of our collective population microbiome. Actually, we came up with this idea over a year, which may have influenced our own gut microbiome. But as it turns out later, actually the proof principle is present in the picture. In a recent paper, actually it was demonstrated that the sewage microbiome reflects that of the human populations. So in this study, actually the authors found that some 15% of these were attributed to the human technical origin. In addition, actually, the distribution of microorganisms reflected the population, which was studied with this microbiome assessment with 81.9% accuracy, which is very good. And the drawback of the current study is that it was done by 16S gene profiling. So in our view, actually, this is just a proof of principle, and actually it should spark, in our opinion, wilder studies where, by assessing the waste of the microbiome, we may learn, and by dynamically assessing the waste of the microbiome, we may extract information for the health, regional health, and population health in dynamics. Actually, we may predict some health issues and possibly this information may be used for prevention. So that's it. Thank you.